Hello friends, welcome to Sam Biology class. Friends, today we shall discuss about the complement system. The term complement refers to a set of serum proteins that cooperate with both the adaptive and the innate immune system to eliminate blood and tissue pathogens. Like the components of the blood clotting system, complement proteins interact with one another in catalytic cascades. Complement proteins play many roles. Various complement components bind and opsonize bacteria, rendering them susceptible to receptor-mediated phagocytosis by macrophages. Other complement proteins elicit inflammatory responses. They also interface with the components of the adaptive immune system and clear immune complexes from the serum. They also kill or eliminate apoptotic cells. Finally, a membrane attack complex is assembled from complement proteins that directly kill pathogen by creating pores in the microbial membrane. The complement system is a complex group of more than 30 glycoproteins which are classified into 7 functional categories. In order to understand the functioning of the complement system, we must know about its components. The first functional category include various initiator complement components. These complement proteins initiate their respective complement cascades by binding with specific soluble and membrane-bound molecules. By binding with their activating ligand, these initiator components undergo a conformational change which also change their biological activity. Example of the initiator components are the C1q complex, mannose binding lectin, and the picolines. The second functional category include enzymatic mediators such as C1R, C1S, MASP2, and factor B. Several components of the complement system are proteolytic enzymes that cleave and activate other members of the complement cascades. Some of the proteolytic enzymes are activated by binding with other macromolecules and undergoing a conformational change. Other proteases are inactive until cleaved by another proteolytic enzyme. There are two enzyme complexes known as C3 convertase and C5 convertase which cleave the complement components C3 and C5 respectively. The third functional category include various membrane binding components known as opsonins. Upon activation of the complement cascades, several complement proteins are cleaved into two fragments, each of which play a specific role. For complement components C3 and C4, the larger fragments are C3b and C4b. Both of them serve as opsonins. They bind to the surface of microorganisms where they act as binding tag for phagocytes, thereby promoting the phagocytosis of various microorganisms. As a general rule, the larger fragments are designated with the suffix b, but there is an exception in case of complement component C2. The larger fragment, which is enzymatically active, is designated with the suffix A. Fourth component include inflammatory mediators. Some of the small complement fragments act as inflammatory mediators. They increase the supply of blood to the area where they are released. They also attract other cells to the site of tissue damage. because such effects are harmful when in excess. Such fragments are called as anaphylotoxins. Examples of inflammatory mediators are C3A, C5A and C4A. The fifth functional category include various membrane attack proteins. In the complement system, finally, a membrane attack complex is assembled from various complement components such as C5b, C6, C7, C8 and multiple copies of C9. All these components are inserted into the microbial cell membrane where they punch holes that resulted in the lysis of the microorganisms. The sixth category include various complement receptor proteins. These proteins are the receptor molecules present on the surface of cell where they bind various complement proteins and signal specific cell function. For example, the complement receptor protein known as CR1 bind to the complement component C3b which is present on the surface of pathogens, thereby triggering their phagocytosis. Another complement receptor is C5AR. Binding of the complement component C5A to C5AR trigger the degranulation of neutrophils and inflammation. Last category of complement components include regulatory components. Host cells are protected from unintended complement-mediated lysis by the presence of regulatory components. These components can be membrane bound or soluble. Example is factor I. Factor I degrade C3b. And another regulatory component is protectin. Protectin inhibit the formation of membrane attack complex. So friends, this is about various components of the complement system. There are three major pathways of complement activation. These pathways are classical pathway, lectin pathway and alternative pathway. This figure shows the major initiation pathway of the complement cascade. Although the initiating events in each of the three pathways are different, all of them leads to the generation of an enzyme complex known as C3 convertase. This enzyme cleaves C3 molecules into two fragments, C3A and C3B. Classical pathway and lectin pathway use the dimer C4B2A for C3 convertase activity, whereas alternative pathway use another complex, that is C3BBB for C3 convertase activity. However, in all these pathways the end result is same, that is there is a dramatic increase in the concentration of C3b molecules. C3b is a centrally located multifunctional complement protein. The second set of convertase enzyme is C5 convertase, which are formed by the addition of C3b component to the C3 convertase. C5 convertase cleave C5 molecules into two fragments, C5A and C5B. C5A is an inflammatory mediator, whereas C5b is the initiating factor of the membrane attack complex. All these pathways are divided into three phases. First phase is initiation, second phase is amplification, and the last phase is termination, in which various activities like inflammation, opsonization and lysis are carried out by activator components of the complement system.